The handouts or review materials must be treated with utmost confidentiality. It shall be the responsibility of the person whose name appears therein that the handouts or review materials are not photocopied or in any way reproduced, shared, or lent to any person or disposed in any manner. Any handouts or review materials found in the possession of another person whose name does not appear therein shall be prima facie evidence of violation of RA 8293. Top-notch review materials are updated every 6 months based on the current trends and feedback. Please buy all recommended review books and other materials.
We will now discuss the next pair of pathways. So this will be for glycogen metabolism. So in glycogen metabolism, we will be discussing glycogenesis, which is the creation of glycogen, and in glycogenolysis, which is the breakdown of glycogen. So just like glycolysis and gluconeogenesis, these are opposite pathways, wherein you tend to do glycogenesis during the fed state and glycogenolysis during the fasting state. So what is glycogen? So glycogen is the major storage carbohydrate in animals. So meaning to say, for example, kanina sabi natin pag kumain ka sa buffet, yung excess glucose, so that excess glucose will not be stored as glucose. To be able to store it, you will store it as glycogen. Is that clear? So the storage form of glucose in animals, including humans, is glycogen. Now, where do we actually store it? We store glycogen in muscle and in the liver. So about 400 grams will be stored in muscle and about 100 grams will be stored in the liver. Now, by percentage weight, no? So if you talk about percentage weight, so actually your liver, it's about 10% glycogen. And muscle, it's about 1 to 2% glycogen. Pero dahil nga mas malaki yung muscle mass natin, kaya you can store more glycogen in the muscle. So magkaiba sila ng reason for storing glycogen. Sa liver, why does the liver store glycogen? The liver stores glycogen to be able to provide glucose to other tissues. So si liver, no? Ano yan, no? Mapagbigay yan, no? Generous, no? So si liver, generous, almost martyr, sabi nga natin, di ba? So magsustorya yan ng glycogen para during periods of fasting, it can distribute glucose to other tissues. Si muscle, yung glycogen na sinustore niya para lang sa sarili niya yon, for its own use. So si muscle, paasa na, selfish pa, no? So yung glycogen, sinustore niya para lang sa sarili niya yan. Now, what makes up glycogen? If you look at glycogen under a microscope, no, pag tinignan mo yung molecular structure niya, glycogen actually consists of alpha-D-glucose. So, paulit-ulit lang, it's a homopolymer, meaning to say all the molecules are glucose. To be specific, alpha-D-glucose. So, these glucose molecules are joined together using glycosidic bonds. Now, there are two types of bonds, alpha-1,4 and alpha-1,6 glycosidic bonds. So ito siya, no? So when you look, no? So this is a glycogen molecule. Para siyang Christmas tree. At the center, you have the glycogenine, which is a protein. And then, may mga nakakabit na glycogen. Okay? Merong mga chains, no? Ito yung mga chains ng glycogen. Okay? So in Lippincott, it says there that every 8 to 10 residues, you get a branch point. Okay? Every 8 to 10 residues, you get a branch point. So this is really the difference between glycogen and starch. No, in starch, there's no branching pattern. No? In starch, there's no branching pattern. So ibig sabihin, no, so you will see here, ito kabit-kabit to na glucose. This is a branch point. And then glucose yan, kabit-kabit. That's a branch point. That's a branch point, kabit-kabit na glucose. So when you try to think about it, bakit ganun? Bakit kailangan yung glycogen mukhang Christmas tree? Bakit hindi na lang isang dire-diretso? No? Bakit kaya? No? So, kunyari, nag-ipon ka ng glucose, kumain ka ng maraming glucose, bakit kailangan ganyan pa? Ba't mo pahirapan yung sarili mo? Ba't hindi mo na lang pagdugtong-dugtongin? So, when you think about it, kunyari, kailangan mo ng glucose. No? So, nag-store ka as glycogen. Kung yung glycogen, ganyan lang, isang straight strand lang, paano ka kukuha ng glucose? two at a time, di ba? Both ends lang, no? So, dalawa lang yung nakukuha natin ng glucose kasi nga straight line siya. Pero kung ganito yung structure ng glycogen, parang Christmas tree, pwede tayong kumuha from all ends. Sabay-sabay, di ba? Sabay-sabay kukuha tayo ng glucose, marami tayong nare-release na glucose agad. That's the reason why we have this structure. Now, as I said, when you try to look at it, ano itsura niya, no? So, you will see here, no, your glycogen, meron tayong linear strand and branch points. And this is very important. When you look at the linear strands, they are joined together by alpha-1 for glycosidic bonds. Alpha-1 for glycosidic bonds. In contrast, the branch points, they're alpha-1, 6. No? They're alpha-1, 6. So, paano natin yun matatandaan, no? So, remember, linear strand, alpha-1, 4. Branch points, alpha-1, 6. So, ito yung mnemonic natin. So, pag sulat tayo ng number 4, no? So, pag magsusulat ka ng number 4, so, dapat ganito na kayo magsulat ng number 4. So, for example, here, yung glucose. No? So, glucose. So, kunyari, pag dudugtong-dugtongin natin itong mga glucose residues na to. Okay? Kung gusto natin silang pagdugtong-dugtongin, okay? Sulat tayo ng number 4. Si number 4, ganyan. Okay? Doon siya nakaturo sa side. No? Si number 4, ganyan. Sa side. No? So, if you look, number 4, doon siya nakaturo. So, alpha-1, 4, sa side. No? Ibig sabihin, the linear strands, alpha-1, 4 yan. So, doon. So, turo mo nga, alpha-1, 4. In contrast, pag gusto natin ng branch point, anong kailangan natin? Alpha-1, 6. So, si 6, ganun. Sa taas. Okay? So, alpha-1, 4, linear strand. Alpha-1, 6, nakaturo sa taas. Okay? So, turo mo nga. Alpha-1, 4, sa side. Alpha-1, 6, sa taas. O, yung hindi magturo, babagsak sa exam. Yung hindi nakaturo, so, nakababagsak sa exam. Dapat ituro mo. Okay, next. So, tatandaan na, anong purpose ng glycogen? Glycogen is the storage form of glucose. Where do we store it? No? So, saan tayo nagsusstore ng glycogen? Sa liver and muscle. So, ito tatandaan, ha? Sabi natin kanina, pag gluconeogenesis. Saan nangyayari ang gluconeogenesis? Sa liver and kidney. Bakit kidney? Kasi gluconeogenesis. In contrast, pag glycogen metabolism, glycogenesis, glycogenolysis, meron bang ni? Wala, di ba? So, dahil wala, it's liver and muscle. Okay, so, glycogen metabolism. Meron bang ni? Wala. So, it will occur where? Liver and muscle. So, always remember that. So, glycogenesis. So, glycogenesis, it's for synthesis of glycogen. So, mag-glycogenesis tayo pag gusto natin gumawa ng glycogen. Let's analyze. Kailan mo gustong gumawa ng glycogen? Pag well-fed or pag fasting? So, syempre, no, pag bagong kain ka, doon tayo magsusstore for future use. Di ba? So, pag bagong kain tayo, doon tayo magsusstore for future use. Kaya, pag well-fed, mataas ang insulin, doon tayo mag-glycogenesis. Where does it occur? In the liver and in the muscle. What part of the cell? Cytosol. So, all the glycogen reactions happen in the cytosol. The substrate is alpha-D-glucose and the product is glycogen. So, essentially, pag sinabi natin glycogenesis, no, yung glucose, gagawin lang natin glycogen. Glucose to glycogen. The rate-limiting step is glycogen synthase, which creates the alpha-1 for glycosidic bond. So, siya yung nagpapahaba ng glycogen chains, no, or glycogen synthase. So, how do we begin, no? So, as you can see here, we begin first, no, with the glycogenine protein. So, you will see here, no, most of the time, ganito kasi nangyayari. Most of the time, meron na tayong, no, kunyari, gusto nating mag-synthesize ng glycogen from scratch. No, from the very beginning. Kailangan magsisimula muna tayo kay glycogenine protein. So yung protein, doon na tayo magsisimula. Tapos yun yung kakabitan natin ng mga glucose molecules. But actually, what happens most of the time is like this. Meron na tayong tira-tirang glycogen. So you see, meron ng tira-tirang glycogen with glycogenine at the center, and then meron na siyang konting nakakabit. Ang ginagawa na lang ng body natin, pahahabain natin siya. Okay? So from this, yung tira-tirang glycogen, papahabain na lang natin. Okay? So yung natira natin from the last time, no? So pahahabain natin. Next. Okay? So paano natin siya pinapahaba? Pinapahaba natin siya by adding glucose residues, no? So dadagdagan natin ng glucose sa dulo. So dun papasok ngayon si UDP glucose. So you see here, this is UDP glucose. So this is the glucose residue. May katabi siyang UDP. Anong role ni UDP? Si UDP para siyang yaya. No? Para siyang yaya. So si yaya, ihahatid niya si glucose dun sa dulo ng glycogen molecule. Okay? We will see this later. So once again, we begin with UDP glucose, which is activated glucose. So paano nangyayari yun? From glucose, kunyari kumain tayo, yung glucose, gagawa tayo ng glucose 6-phosphate. Glucose to glucose 6-phosphate, that's the first step of glycolysis. And then that glucose is converted to glucose 1-phosphate. And then glucose 1-phosphate is converted to UDP glucose. This is activated glucose. Okay, so doon ang gagaling si UDP glucose. Does that make sense? No, yun yung first step natin, no? Yung glucose to glucose 6-phosphate galing glycolysis. Yes. Kasi nga sabi natin, kapag well-fed tayo, active din yung glycolysis. Pinophosphorylate natin yung glucose. Sino kaya yung magpo-phosphorylate kay glucose? Si glucokinase or si hexokinase? Balikan natin kanina. Sabi natin, no? Glucokinase and hexokinase, they can both phosphorylate glucose. Si hexokinase, nakikita most tissues. Si glucokinase, found in the liver and pancreas. Magkaiba sila ng purpose. Di ba? Anong purpose ni hexokinase? To meet the cell's need. Anong purpose ni glucokinase? To store for future use. So to answer my question, ang magko-convert ng glucose to glucose 6-phosphate in the liver for storage as glycogen ay si glucokinase. Okay. Eventually, nakakagawa na tayo ng mga UDP glucose. Anong purpose ng UDP glucose? It's activated glucose. Si UDP, iyahatid niya si glucose. So you will see, let's look here. So kunyari ito yung dulo ng glycogen, no? So this is the end of the glycogen strand. Sabi natin may glycogen, kailangan nating pahabain. So what will happen is this. So this is UDP glucose. Si yaya, iyahatid niya ngayon si glucose. And then using glycogen synthase, look what will happen. At the non-reducing end of glycogen, kakabit si glucose, no? So si glucose ikakabit doon. Using the enzyme glycogen synthase, you synthesize, you create what bond? Side. Alpha 1,4. Nasaan na si yaya? Umuwi na, no? So si yaya, iyahatid lang niya si glucose, no? Kakabit lang niya si glucose, tas uuwi na din siya. Hindi siya sasama dun sa glycogen molecule. Okay, so essentially, yung UDP, ang purpose lang niya to activate glucose, no? Kasi if you don't activate it, hindi siya basta-basta didikit. So, ganun lang. Kabit lang tayo ng kabit ng glucose using UDP glucose. Humahaba ng humahaba yung glycogen strand. So, as you can see, pahaba ng pahaba yung glycogen strand. Gusto ba natin yon? Ayaw. Kasi ayaw natin ng mahaba lang yung glycogen, di ba? Anong gusto natin gawin? Gusto natin magkaroon ng branch point. So, ang nagpapahaba, glycogen synthase. What will happen next? We need to create branches. And we will do this using the branching enzyme. So the branching enzyme, what will it do? It will cut this off and transfer to create the alpha 1, 6, nakaturo sa taas. Okay? So ano na nangyari? Humaba na siya, nag-create na tayo ng branch. What happens next? Pahahabain na naman natin ulit. Habain na naman. So you see here, papahabain na naman natin yan. No? What enzyme will create more bonds? Glycogen synthase. Sino maghahatid kay glucose? Si UDP, ang kanyang yaya, UDP glucose. Pag mahaba na, sinong tutulong kay glycogen synthase? Si the branching enzyme, which will create the alpha 1,6 bond. By repeating that process over and over, lalago ngayon yung glycogen molecule natin. Okay? Lalago yung glycogen molecule natin. So from the glycogen primer, malaki na ulit siya, ready for future use. Again, glycogenesis. Glycogenesis active during fed state. What is elevated? Which hormone? Insulin. Okay? What about glycogenolysis? So when we say glycogenolysis, breakdown of glycogen, no? So kailan naman kaya natin gustong mag-breakdown? When do we want to mobilize stored glycogen? Well-fed or fasting? Fasting, no? Kasi essentially here in glycogenolysis, yung glycogen, ibabalik na natin siya sa glucose. Bakit kaya natin kailangan siyang ibalik sa glucose? Kasi hindi ka na kumakain, no? Kasi wala ka ng glucose kasi hindi ka na kumain, no? 'Di ba kanina no nag-scrub in ka sa Whipples. Tapos ang tagal-tagal hindi pa matapos yung residente, no? Kailangan mo na ng glucose to provide your tissues. So what you can do, yung glycogen that we stored in your liver and muscle, we can convert that back to glucose to be distributed to the other tissues or to be used by the muscle itself. So glycogenolysis therefore is active during fasting, and it is activated by glucagon, inhibited by insulin. So just like glycogenesis. So glycogenolysis, meron bang ni? Wala. So it occurs in the liver and muscle. Once again, it occurs in the cytosol. Substrate, glycogen, product, glucose or glucose 6-phosphate. I will explain later. The rate-limiting step is glycogen phosphorylase. So this is the opposite of glycogen synthase. So yung glycogenesis and glycogenolysis, even though they're opposite pathways, they utilize entirely different enzymes. So unlike glycolysis and gluconeogenesis that use some similar enzymes, yung glycogenesis and glycogenolysis magkaiba talaga. So ano nangyayari? So imagine the glycogen chain. So you see here, this is the glycogen chain. Okay? So this is the ito yung dulo ng glycogen. Mahaba yung glycogen. Okay? Kailangan nating gamitin na yung glucose. Ang gagawin, no, first we need to shorten it. So using the enzyme glycogen phosphorylase. So ito yung dulo ng glycogen, ito si glycogen phosphorylase. Anong gagawin niya? Kakabitan niya ng phosphate, kaya siya phosphorylase. Kakabitan niya ng phosphate, tapos kakagatin. Okay? Kabit ng phosphate, kagat. Kabit ng phosphate, kagat. Kabit ng phosphate, kagat. So, kagat, 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 kagat. So, ano mangyayari? Parang ganito. Kinakagat niya yung dulo ng glycogen strand. Sino yan? Hindi si Pac-Man, no? Yan si glycogen phosphorylase. Now, what you need to remember about glycogen phosphorylase, it requires vitamin B6 or pyridoxal phosphate. Okay, so kagat, 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 kagat. What bond does it cleave? Alpha 1,4, yung linear strands. However, pag-apat na lang, no? Pag-apat na lang, so kagat, 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 kagat. Pag-apat na lang, anong mangyayari? Sasabihin niya, ay, ayoko na. Hindi niya inuubos, no? So, kagat, 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 kagat. Pag-apat na lang, that's what you call your limit dextrin. Titigil na siya. Papasok naman yung next enzyme. The next enzyme is called your debranching enzyme. So, the debranching enzyme, what will it do? Yung tatlo, ililipat niya. So, from here, yan yung tinatawag natin na limit dextrin. Ililipat niya yung tatlo. Okay? Ililipat niya yung tatlo using its alpha 1,4 glucan transferase. Okay? And then, yung nag-iisa, okay? Yung nag-iisa, gugupitin niya yan. Okay? Using its alpha 1,6 glucosidase, gugupitin niya yan, free glucose. Nakalaya na si glucose. Okay? So, mahaba na ulit. Okay? So, ulit. So, again, anong mangyayari? So, if we repeat that again, initially, si glycogen phosphorylase, kagat, 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 kagat. Bago kagatin, kakabitan mo niya na ng phosphate. Kaya look, the product of glycogen phosphorylase is glucose 1-phosphate. Kailan siya titigil? Pag-apat na lang, that's what we call your limit dextrin. Pag limit dextrin na, anong mangyayari? No? Pag limit dextrin na, papasok si debranching enzyme. Si debranching enzyme, ililipat yung tatlo. Tapos yung natitira, tatanggalin niya. So therefore, it is your debranching enzyme that cleaves your free glucose alpha-1,6 bond. So if they ask in the board exam, no, which enzyme will cleave alpha-1,6? The correct answer is your debranching enzyme. Which enzyme will cleave alpha-1,4? So it's your glycogen phosphorylase, pati si debranching. Kasi remember, ginawa natin si debranching yung maglilipat ng limit dextrin, di ba? So pag ganyan, glycogen phosphorylase ulit. So mahaba na naman, pwede na naman ulit siyang kagatin. Kagakagatin na naman niya, tapos titigil na naman siya pag 4 na lang yung natira. Paulit, ulit, ulit. No. Ano mangyayari doon sa mga glucose 1-phosphate? Those glucose 1-phosphate molecules that you see in the liver, they are converted to glucose 6-phosphate. And then glucose 6-phosphate, okay? Glucose 6-phosphate, tatanggalin natin yung phosphate. Bakit kailangan tanggalin yung phosphate? Para makalabas si glucose. Diba sabi ko sa inyo kanina, no? We need to remove phosphate using glucose 6-phosphatase. If you don't remove the phosphate, what will happen? Your glucose will not be able to go out of the liver tissue. Hindi niya mapapamigay yung glucose. Now, in the muscle, it's different. In the muscle, yung glucose 6-phosphate, hindi niya na tatanggalin yung phosphate. No? What happens in the muscle, that glucose 6-phosphate, idediretso na niya sa glycolysis. So, doon nagkakaiba si liver tsaka si muscle. So, you will see here, the end product of glycogenesis in the liver is glucose. Kasi nga, anong gusto niyang gawin? It wants to break down glycogen so that it can be distributed to other tissues. Para sabi niya, naku, kawawa naman si brain, wala ng glucose. Kawawa naman si heart, wala ng glucose. Kawawa naman si red blood cells, si kidney. Bigyan ko kaya sila ng glucose. Kaya nga si liver ay martyr. No? Ito naman si muscle, yung glycogen niya, i-breakdown niya, hindi niya tatanggalin yung phosphate. Sabi niya, bakit ko tatanggalin yung phosphate? Ako nag-ipon ito. No? So, si muscle, you will see glucose, the end product is glucose 6-phosphate. Why? Because the muscle does not have the enzyme glucose 6-phosphatase. So, yung glucose 6-phosphate na yun, idediretso na niya sa glycolysis. So, remember this, okay? If they ask you, what's the end product of glycogenolysis in the liver? It's glucose. In the muscle, it's glucose 6-phosphate. And the reason is that muscle does not have glucose 6-phosphatase. And they have different reasons for storing glycogen. The liver stores glycogen for distribution to other organs. The muscle stores glycogen for its own use. Now, sabi natin, di ba? So, the glycogen metabolism happens in the cytosol. However, you can also degrade glycogen in the lysosomes. So, if you remember still in biology, what are the lysosomes? These are what we call suicide bags. So, yung lysosomes, these are organelles in your cells. No? They can break down complex molecules. So, one such complex molecule will be glycogen. So, your glycogen can be broken down in lysosomes using lysosomal acid maltase. So, this degrades about 1 to 3% of glycogen. We don't really know the reason why this is there, no? But this is clinically significant. Why? If you do not have lysosomal acid maltase, what will happen? Glycogen will accumulate. And where will it accumulate? It will accumulate in the lysosomes. No? It will accumulate in the lysosomes. So, at yun yung nangyayari saan? Sa Pompe disease. So, in Pompe disease, dahil walang lysosomal acid maltase, tataas ngayon yung glycogen sa lysosomes. Now, let's take a look here. No? So, this table summarizes glycogen metabolism. No? So, this table summarizes glycogen metabolism. What you can see here, the first column will be glycogenesis. The second column will be glycogenolysis. Okay? So, if you are in the fed state, so let's analyze this again. Don't just memorize this table. No? Dapat, ang goal natin, you need to be able to answer this without memorizing the table. Simple lang naman siya, di ba? So, glycogenesis, the enzyme is glycogen synthase. Glycogenolysis, the enzyme is glycogen phosphorylase. Well, let's try to see. So, kunyari, no? Kung maraming glucose, no? Maraming glucose 6-phosphate. No? So, kung maraming glucose 6-phosphate, anong i-activate mo? Glycogenesis or glycogenolysis? Of course, glycogenesis. Gagawa tayo ng glycogen. Okay? I-inhibit natin yung glycogenolysis. Kasi marami ng glucose eh. Hindi mo na kailangan gumawa ng glucose. O, sa muscle, kapag may calcium, ano ibig sabihin nun? Pag may calcium, yung muscle nag-exercise, di ba? Kailangan niya mag-contract. Kailangan kaya niya ng energy? Yes, di ba? So therefore, anong gagawin mo? Isu-store mo ba yung glucose or i-breakdown mo ba yung glycogen? So dahil kailangan natin ng energy, anong kailangan mong gawin? Yung glycogen sa muscle, bini-breakdown natin yan. So that's why, for example, tatakbo ka ng 10 kilometers or half marathon, you will break down your glycogen molecules so that your muscles have glucose for energy. Hormones. Insulin. Kapag mataas ang insulin, anong i-activate mo? Glycogenesis or glycogenolysis? So, insulin, you are well fed. Magsustor ka ba ng glycogen? Magbe-breakdown. Eh di magsustor, no? Then you will store glycogen. In contrast, pag mataas ang glucagon, what will you do? Okay? You will break down glycogen. Kasi pag mataas ang glucagon, you are fasting. No? You are fasting. Therefore, if you are fasting, then what do you need to do? If you are fasting, you need to break down glycogen so you will have a source of glucose. Now, you will see there phosphorylation, dephosphorylation. We will discuss this later. No? We will discuss this later. Tatandaan lang natin, di ba? Ano ulit yung phosphate? Para siyang on and off switch. No? Para siyang on and off switch. And I will give you later, no, an example on how we remember this best. So, you will see here, no, in glycogen metabolism, no, insulin and glucagon. So, pag insulin, no, what will insulin activate? Insulin will activate glycogenesis. No? So, insulin is high, it will activate glycogenesis. When glucagon is high, it will activate glycogenolysis or the breakdown of glycogen. So, clinical correlates. So, for glycogen metabolism, here's what you need to remember. We have what is known as your glycogen storage diseases. So, ano nangyayari sa glycogen storage diseases? Sa glycogen storage disease, may enzyme sa glycogenolysis na deficient or missing. No? So, we have deficient enzymes for glycogenolysis. So, ibig sabihin yung glycogen, maiipon siya. Hindi natin siya ma-breakdown to glucose. So, you have elevated enzymes. No? So, you have deficient enzymes. So, we cannot break down glycogen. So, glycogen will tend to accumulate. And where will it accumulate? Either in the muscle or in the liver. No? So you have accumulation of glycogen within cells. So this table, no, let's try it again. No? Sabi ko sa inyo, don't just memorize. Let's try to see what is happening. So ito, no, ang very important, you have very different types of glycogen storage diseases, but the most important ones are type 1, type 2, type 3, and type 5, no? So, type 1, type 2, type 3, and type 5. So, ang mnemonic dyan, very poor carbohydrate metabolism. Pero one time, ang tinanong nila, HERS, type 6, no? So, kabisaduhin nyo na lang din lahat, no? So, okay, let's try to see what happens. So, type 1 is known as von Gerdt disease. In von Gerdt disease, you do not have glucose 6-phosphatase. Well, let's analyze. Kung wala tayong glucose 6-phosphatase, ano yung mangyayari? So, this is von Gerdt. Imagine, kung walang glucose 6-phosphatase, anong nangyayari? We cannot convert glucose 6-phosphate to glucose. If you cannot convert glucose 6-phosphate to glucose, anong sabi natin? Kung hindi mo kayang tanggalin si phosphate, si glucose, hindi siya makakalabas. No? So, pag binreakdown natin yung glycogen, hindi nakakalabas si glucose. That's why, when you go on a fasting state, ano nangyayari? Si liver, hindi niya ma-release yung glucose. You become hypoglycemic. So you will see here, the liver, there will be accumulation of glycogen. Kasi nga hindi niya nabe-breakdown ng maayos yung glycogen and you will have fasting hypoglycemia. You will see in the cytosol, maraming glycogen. Okay, does that make sense? So dahil hindi natin ma-breakdown si glycogen. Now, compare it with McArdle syndrome. So si McArdle syndrome, ang wala naman sa kanya, muscle phosphorylase. So wala siyang muscle phosphorylase. Ano ibig sabihin nun? Yung skeletal muscle niya, hindi niya makagat-kagat yung glycogen. Ang manifestation nito will be cramps and myoglobinuria. No? Myoglobin will be broken down kasi mamamatay yung muscle cells. No? And they will release myoglobin into the circulation. Bakit walang hypoglycemia? Because remember, si muscle, hindi naman talaga siya nagpo-provide ng glucose to other tissues, di ba? Hindi naman talaga siya nagpo-provide ng glucose to other tissues. So, nag-store siya ng glycogen, pero hindi niya magamit. No? Buti nga. So, but what will happen is, they will have accumulation of muscle glycogen, and your muscle will have cramps and myoglobinuria when you have exercise. Now, Cori disease is just like von Gurk, pero mas mild. Kasi when you look, ano ba yung wala sa Cori? Ang wala sa Cori disease yung debranching enzyme. Meaning to say, nakakakuha pa tayo ng glucose sa glycogen, pero pag limit dextrin na, tigil na. So, hindi natin nauubos yung glycogen. So, sayang. No? May natitirang limit dextrin. That's why in Cori disease, even though you have hypoglycemia, it's milder, and you have elevated limit dextrin. So, what about Hirsch disease? Yung Hirsch disease, para siyang McArdle. Pero this time, sino walang phosphorylase? Si liver. So, kung walang phosphorylase si liver, so si liver, hindi niya makagat-kagat yung glycogen. Ano mangyayari sa'yo again? Again, hypoglycemia and hepatomegaly. Kasi nga, hindi mo makagat yung glycogen, maiipon lang siya sa liver. And then finally, this is pump disease. So, pump disease, ang mnemonic lang dito, pump affects the pump of the body. So, the enzyme deficiency here is lysosomal acid maltase. Meaning to say, you do not have acid maltase in the lysosomes. So, you will have accumulation of glycogen in the lysosomes. And in infants, this manifests as cardiomegaly, heart failure. So, you see here, you have glycogen accumulation in heart muscle, you have cardiomegaly. So, treatment, you replace the enzyme. No? So, you replace the enzyme. They need to receive enzyme replacement. Otherwise, so you see here, after enzyme replacement, nag-reverse yung symptoms. No? In adults, hindi lang siya heart failure, they can also manifest para siyang muscle dystrophy. No? So kapag juvenile onset, hypotonia, heart failure. Pero kapag adult onset, ang presentation niyan, muscle dystrophy. Okay? So again, just remember, glycogen storage diseases may enzyme deficiency, and because of these enzyme deficiencies, glycogen will accumulate in the tissues. Okay, so before we move forward, so let me just direct your attention to this row. No? So you will see here, no, effect of phosphorylation. So you will see here, if glycogen synthase is phosphorylated, it's inactive. Pero si glycogen phosphorylase naman, baligtad. If you phosphorylate it, it becomes active. If you dephosphorylate glycogen synthase, it becomes active. And if you dephosphorylate glycogen phosphorylase, it's inactive. So, paano nyo i-memorize siya? Nakakalito, di ba? Parang 1010001111, no? So, paano natin maaalala yan, no? So, how do we best remember this? So, hindi lang to dito sa glycogen metabolism, but for all the other phosphorylation questions. So, let's go back. What did I tell you earlier? Sabi natin kanina, okay? So, sabi natin kanina, your phosphate is like a switch. So, kunyari, meron tayong enzyme, no? Okay, we have this enzyme. Okay? Pag kinabitan natin siya ng phosphate, it will either be switched on or switched off. So, sino magkakabit ng phosphate? No, usually, kinase, no? So, protein kinase A or protein kinase C. So, paano natin ngayon malalaman pag kinabitan ba natin ito ng phosphate? Ma-activate ba siya or ma-inhibit? So, ito yung tatandaan nyo, no? Ito lang yung tatandaan nyo. Kapag well-fed, so tandaan nyo, well-fed, okay? So, pag sinabi natin well-fed, so well-fed, okay? Pag well-fed, walang phosphate. Okay? Well-fed, walang phosphate. So, dephosphorylated tayo. Okay? Pag fed state, okay? Kapag fasting, no? Kapag fasting naman, no? So, well-fed, walang phosphate. Pag fasting, phosphorylated. Okay, huwag niyong kakalimutan, no? So, well-fed, walang phosphate. Fasting, phosphorylated. Paano nangyari yun, no? Yun yung una niyong tatandaan. Kasi remember, here, no? So, kapag meron tayong glucagon, so glucagon, when it binds to its receptor, it activates adenylyl cyclase, which converts ATP to cyclic AMP. And then cyclic AMP, what will it do? It activates protein kinase A. So si protein kinase A, anong ginagawa niya? Pinophosphorylate niya yung mga enzymes. Kaya nga pag may glucagon, fasting, phosphorylated. In contrast, pag insulin, well-fed, walang phosphate. Okay? So glucagon, fasting, phosphorylated. Insulin, well-fed, walang phosphate. So how will it help you? So, for example, tingin lang kayo sa akin. If I ask you, what will happen if you phosphorylate glycogen phosphorylase? So don't look at the table, just look at me. If you phosphorylate glycogen phosphorylase. So isip tayo ngayon. Pag sinabi niyang glycogen phosphorylase, ano bang pathway si glycogen phosphorylase? Glycogenolysis, di ba? Kagat, no? So glycogenolysis. Pag phosphorylated, ano ibig sabihin nun? Phosphorylated fasting, di ba? Phosphorylated fasting. So pag fasting, anong gusto mong gawin sa glycogenolysis? No, pag fasting ka, gusto mo bang mag-breakdown or mag-synthesize ng glycogen? You want to synthesize. You want to break down, no? Gusto mong i-break down kasi nga fasting ka. So therefore, anong sagot? So kung phosphorylated glycogen phosphorylase, it will be active. Okay? So if you phosphorylate glycogen phosphorylase, it will be active. Let's try again. What will happen if you dephosphorylate glycogen synthase? So what will happen if you dephosphorylate glycogen synthase? Let's try to think. Pag dephosphorylate, walang phosphate. Walang phosphate, therefore, well-fed. Well-fed, walang phosphate. Para saan ba yung glycogen synthase? It's for synthesis of glycogen. Diba? Glycogenesis. So kung well-fed ka, gusto mo bang mag-synthesize ng glycogen? Yes. So that's why pag walang phosphate, you're in the well-fed state and you want to activate glycogen synthase. So hindi lang to dito, no? Hindi lang to dito. So pwede rin to sa ibang enzymes. So for example, pyruvate kinase. What will happen if you dephosphorylate pyruvate kinase? So mag-isip tayo. Ang pyruvate kinase, saang pathway ba si pyruvate kinase? No? Saang pathway ba si pyruvate kinase? Sa glycolysis. No? So kung dephosphorylated, walang phosphate. Well-fed, walang phosphate. In the well-fed state, mag-glycolysis ka ba? Okay? Mag-glycolysis ka ba? The correct answer is yes. That's why dephosphorylation activates pyruvate kinase. Dephosphorylation activates pyruvate kinase. One more. What will happen if you phosphorylate phosphofructokinase 1? So phosphofructokinase 1 is part of glycolysis. Kung phosphorylated, may nakakabit na phosphate. Fasting. So in the fasting state, mag-glycolysis ka ba? No. So that's why in the fasting state, okay, in the phosphorylated state, phosphofructokinase 1 is inhibited. So I know it will take a bit of time, no? So you need to practice on this, no? So just review, no? But yun yung basic principle. Tatandaan nyo lang, pag walang phosphate, so walang phosphate, WF, well-fed, mataas ang insulin. Kapag phosphorylated, may phosphate, fasting yun, no? And it's because of glucagon. Okay? So, finally, let's review the 4 Gs. So, nakapag-aral na tayo ng 4 Gs. So, in summary, let's summarize the 4 Gs. So, if we summarize the 4 Gs, no, you have two pairs of opposite pathways. Glycolysis and gluconeogenesis, glycogenesis and glycogenolysis. So, let's see, no? Which G happens in all cells? No, which G happens in all tissues? Answer is glycolysis. Which 2 Gs happen when you are well-fed? No, so which 2 Gs will happen when you are well-fed? Glycolysis, which is the breakdown of glucose, and glycogenesis, which is the synthesis of glycogen. Which 2 Gs will happen when you are fasting? So, isipin mo lang sa pair number 1, it will be gluconeogenesis. Kasi gagawa ka ng glucose. Sa pair number 2, it will be glycogenolysis. I-breakdown mo yung stored glycogen mo para makakuha ka ng glucose. Diba? Oh. Which G happens in both cytosol and mitochondria? Okay. Gluconeogenesis. No? Gluconeogenesis. Which G happens in the liver and the kidney? Okay, so liver and kidney. Okay, liver and kidney only. Gluconeogenesis. Which happens in the liver and the muscle? Glycogenesis and glycogenolysis. Okay. Which Gs will be activated by insulin? Glycolysis and glycogenesis. Which Gs will be activated by glucagon? Gluconeogenesis and glycogenolysis. O, di ba? Madali lang. Okay? The next step will be for you to memorize the important enzymes in these pathways. So after discussing the major pathways of carbohydrate metabolism, we will now go to the other important carbohydrate pathways. So here we will discuss the metabolism of other hexoses, specifically galactose and fructose. We also learn about sorbitol, your acidic sugars, and then finally we will discuss the pentose phosphate pathway. So galactose and fructose. If you recall, both galactose and fructose are also monosaccharides, and they also play an important role in our diet. So in here, this diagram shows you. So here at the center, we have glycolysis. No? So this diagram tells you how does galactose become an energy source for the body. So galactose metabolism will focus first on the right side. So briefly, as you can see here, galactose is converted to galactose 1-phosphate, and then galactose 1-phosphate is converted to UDP galactose, which will then be converted to UDP glucose and then glucose 1-phosphate, entering now as glucose 6-phosphate. So the entry point of galactose is glucose 6-phosphate, as you can see here, and now it will be able to go down to pyruvate in aerobic glycolysis. So galactose metabolism. So galactose is found in lactose, so milk and dairy products. The first step is the phosphorylation of galactose. So galactose is phosphorylated using galactokinase. So galactose to galactose 1-phosphate. And then afterward, as I've said, it is converted to UDP galactose. So UDP galactose, ito yun. No? This is activated galactose. So this one is actually the important step. So ito yun, nandito yun. So dito yung galactokinase, the formation of galactose 1-phosphate. This is the one that forms UDP galactose. The enzyme here is galactose 1-phosphate uridyl transferase. In short, GALT, G-A-L-T. No? So, yun yung pinaka-important dyan. So, step number 2. And then afterwards, as I've said, mako-convert na siya eventually hanggang maging glucose 6-phosphate siya. So, in galactose metabolism, what will happen if you don't have galactokinase? What will happen if you don't have galactose 1-phosphate uridyl transferase? No? If you do not have galactokinase, so step number 1, you will not be able to convert galactose to galactose 1-phosphate using galactokinase. However, pwede pa rin yun gawin ni hexokinase. So remember, no, pwede pa rin yung gawin ni hexokinase. The symptoms of galactokinase deficiency are relatively mild. Meaning to say, you will have elevated galactose in the blood, so galactosemia, and you will have galactose in the urine, galactosuria. Later on, in childhood, they might develop cataracts. Why? Because galactose will be converted to its sugar alcohol, galactitol, and that will cause accumulation of water in the lens, leading to formation of cataracts. So it's relatively mild. Sino yung mas toxic? If step number 2 is deficient. So if you have deficiency of galactose 1-phosphate uridyl transferase, you will have more severe symptoms, and this is classic galactosemia. No? So classic galactosemia, this is present in about 1 in 30,000 births. This is present in about 1 in 30,000 births. And it will present with galactosemia, galactosuria. You will see not just cataract. First few days of life pa lang may cataracts na. The diarrhea, vomiting, and jaundice. If this is not treated, the child will definitely develop severe mental retardation. You'll also have liver damage. Lagi nilang binabanggit sa board exam, no? They will have premature ovarian failure in females. So what's the treatment for both conditions? You need to avoid galactose from the diet. So you remove milk, all dairy products. So not just galactose, but also lactose. So not just galactose, but also lactose. So again, always remember, if step number 1 is deficient, galactokinase, the symptoms are mild. If step number 2 is deficient, then the symptoms are more severe. Now, we go to the next part. The next part naman, we look at fructose metabolism. Yun naman yung nasa left side. Paano naman pumapasok si fructose? You need to remember that between fructose and glucose, no, in the intestines, it is easier to absorb fructose. Why? Because it's just facilitated diffusion. And then, when glucose enters the cell, it does not need insulin, no, unlike glucose, no. So what happens? So when you have fructose, you will see here again fructose converted to fructose 1-phosphate, and then fructose 1-phosphate, it is cleaved into glyceraldehyde and dihydroxyacetone phosphate or DHAP. So yung pasok ni fructose dito sa DHAP and glyceraldehyde 3-phosphate. Okay, this is catalyzed by aldolase B. So, aldolase B, kasi yung aldolase A, yun yung nandito sa glycolysis. Okay? So, that's why fructose metabolism is faster because it will bypass the rate-limiting step of glycolysis, yung PFK1. So, remember, PFK1 is here, nababypass natin yun. So, again, 2 enzymes, fructokinase, which converts fructose to fructose 1-phosphate, and then aldolase B, which cleaves fructose 1-phosphate into glyceraldehyde and DHAP. Right? So, you will see ito yung aldolase B. Oh. Aldolase B, yung fructose 1-phosphate, gagawin niyang glyceraldehyde and DHAP. And DHAP. So, now, okay, paano naman yung deficiencies? No? So, again, if you have deficiency in step number 1, if step number 1 is deficient, that's what you call fructosuria. So in fructosuria, it's so fructokinase deficiency, it's benign and asymptomatic. So these patients, they will just have fructose in their urine, but they're relatively okay. In contrast, if you have aldolase B deficiency, no, so this is what you call hereditary fructose intolerance. No? So what happens? These children, if you start giving them fruit juices, no, especially when you wean them from milk, so they will have severe hypoglycemia, especially after consumption of fructose or sucrose. Bakit ganon? No? Ang nangyayari kasi si fructose, no? Si fructose, aagawin niya lahat ng phosphate. Kukunin niya lahat ng phosphate. It acts like a phosphate sink. No? It acts like a phosphate sink. So fructose 1-phosphate accumulates. No? And then what will happen? It will inhibit, no? So ang mangyayari dyan, no, it will inhibit the enzymes of gluconeogenesis, no? So it will impair gluconeogenesis. So kasi nga the liver will run out of ATP. So at the same time, you'll see, no, you will have jaundice, hemorrhage, hepatomegaly, liver failure, no? So kapag ganun, no, what's the treatment? You need to eliminate all sources of fructose and sucrose in the diet. Okay? So again, galactose and fructose. So remember, galactose enters via glucose 6-phosphate. So dito siya papasok, glucose 1, glucose 6-phosphate, whereas fructose enters via glyceraldehyde 3-phosphate and DHAP. So step number 1 for both conditions, pag walang galactokinase or fructokinase, relatively mild symptoms. In fact, it is asymptomatic in fructokinase deficiency. But if step number 2 is deficient, so that's classic galactosemia. In galactose 1-phosphate uridyl transferase deficiency. For fructose aldolase B deficiency, that's fructose intolerance. So for both conditions, the treatment will be to remove the offending sugar. No? So tanggalin mo yung galactose and lactose, tanggalin mo yung fructose and sucrose. Okay, so this is the summary. So what else do you need to remember? So you need to remember, no? So saan sila pumapasok, ano yung treatment, and then these are usually autosomal recessive conditions, no? So these are autosomal recessive conditions. Okay. So what about sorbitol? So sorbitol, saan galing yung sorbitol? Sorbitol, you must remember, comes from glucose, no? So yung sorbitol galing yan sa glucose. Glucose is converted to sorbitol using aldose reductase. So remember, sugar alcohol, reduction. So glucose to sorbitol, that's reduction. Where does this happen? It happens in these organs enumerated here. Now, no, in liver, ovaries, and seminal vesicles, that sorbitol will be converted to fructose. No? So yung sorbitol na yon, mako-convert yan to fructose. Anong ibig sabihin? In the lens, retina, Schwann cells, and kidney, walang conversion to fructose na nangyari. They remain sorbitol. And this becomes problematic if you have diabetes. If you have diabetes, what happens? Laging mataas yung glucose. If you always have elevated glucose, that glucose will enter these tissues, lens, retina, Schwann cells, kidneys, and that will be converted to sorbitol. O ano sabi natin? Sorbitol is a sugar alcohol. It will attract water. So yung lens, mapupuno siya ng water. You now form cataracts. Yung retina, mapupuno siya ng water. Diabetic retinopathy. Schwann cells, diabetic neuropathy. Kidney, diabetic nephropathy. So kasalanan nyo ni sorbitol. Sorbitol is responsible for the pathophysiology of most symptoms of the end organ damage seen in diabetes mellitus, no? So you must remember that it is the accumulation of sorbitol attracting water that causes these clinical manifestations. Okay, so now, no, so remember this, no? So ano lang yung tatandaan niyo sa mga yan, no? So in the board exam, no? So titingnan niyo lagi yung symptomatology, no? So kapag diabetes symptoms, sorbitol yan. No? Kapag ang symptoms naman niya cataracts, buzzword yan for galactose metabolism, no? So galactose metabolism disorder. Kapag yung symptoms nagsimula nung pinakain ng breast milk or pinakain ng breast milk, galactose yan, no? In fact, galactosemia is one of the few absolute contraindications to breastfeeding. Pero pag yung symptoms nagsimula nung pinakain ng fruit sugars, fruit juices, no? Yun naman fructose metabolism yun. So yun yung mga clues. Abnormal disaccharide digestion. So when we say abnormal disaccharide digestion, dalawa lang lagi yan ang naisip natin. First is lactose intolerance. What is lactose intolerance? If you are lactose intolerant, you get flatulence and diarrhea after ingestion of dairy products. No? This is seen in up to 90% of adults, especially Asians. No? So ano nangyayari? Why do you get flatulence and why do you get diarrhea? Kasi nga, if you consume products that contain lactose, you don't have lactase. Lactase breaks down lactose into galactose and glucose. Kung hindi natin siya ma-breakdown, what happens? That lactose travels down, no? So that acts as an osmotic it acts as an osmotic laxative. It will attract water, causing diarrhea. Bakit ka naman magkakaroon ng flatulence? Because that lactose, pagdating niya sa intestines, sa large intestines, you have bacteria there that will digest the lactose and produce gas. So what's the treatment? Well, you have to avoid dairy products, or you can now consume lactose-free milk. No, meron ng lactose-free milk. Why? That's milk with added lactase already. Or you can take lactase pills, meaning to say, before you consume lactose or dairy products, you take these lactase pills, which will provide the enzyme to digest your lactose. And then you have your isomaltase sucrase deficiency, your sucrase isomaltase complex deficiency. Ito naman, hindi naman to sa Asians. This is found in the Inuit people or the Greenland Eskimos, about 10% of them. Similar manifestation ang hindi naman nila ma-digest, isomaltase and sucrase. So you have to restrict sucrose in these people, among these people, because in these patients, because they will not be able to digest sucrose. So earlier, when we said we convert glucose to sorbitol, that's oxidation or reduction? Reduction. Ngayon naman, pupunta naman tayo sa oxidative pathways. So sa oxidative pathways, we talk about two pathways, the uronic acid pathway and the pentose phosphate pathway. In the uronic acid pathway, this is how we convert glucose to its acidic sugar. So the acidic sugar is glucuronic acid or iduronic acid. So it's the main pathway for producing glucuronic acid and iduronic acid. In some mammals, it is also the pathway for synthesizing ascorbic acid. So it's not possible for guinea pigs and primates like us, because we do not have the enzyme L-gulonolactone oxidase. So if you see here, this is glucuronic acid. So it's glucose, So sorry, this is glucuronic acid. So yung glucose siya pero may extra carboxylic acid. Okay, that's glucuronic acid. Bakit important yung glucuronic acid? Glucuronic acid is produced in the uronic acid pathway. We need this for your glycosaminoglycans, your complex molecules. So yung iduronic acid at glucuronic acid, so they contain a lot of negative charges. They will attract water. And that's an important function of your glycosaminoglycans. They attract water in the extracellular matrix for lubrication, for transport, and everything else. At the same time, glucuronic acid, it's very polar. So for example, if you want to metabolize bilirubin, what you can do, bilirubin is very non-polar. It's very non-polar. So yung glucuronic acid is very polar. Marami siyang negative charge. So pag si bilirubin na non-polar, hydrophobic, kinabitan mo siya ng glucuronic acid, nagiging mas soluble na siya sa water. It becomes more soluble. So it helps in detoxifying insoluble compounds. So bilirubin, steroids, morphine, and other drugs. Okay, so tatandaan nyo lang, uronic acid pathway, glucose to glucuronic acid or iduronic acid. Important for glycosaminoglycans and metabolism of compounds. Finally, we have what is called essential pentosuria. In essential pentosuria, you cannot convert, you have cellulose reductase deficiency. In cellulose reductase deficiency, you cannot convert cellulose to xylitol. Wala naman to, it's not clinical significant, baka lang tanungin sa board, this is in Harper. You have cellulose in the urine, umiihi ka ng pentose, kaya siya essential pentosuria, you excrete excessive amounts of cellulose. This is benign with no clinical consequence. Now take note, we cannot produce ascorbic acid because we do not have this enzyme, your L-gulonolactone oxidase. Right? So the last carbohydrate pathway we will discuss is the pentose phosphate pathway. This is also known as the hexose monophosphate shunt. Now, here's the thing. So unlike the previous pathways we've discussed, wala itong kinalaman sa metabolism. So you do not get energy from this. This does not require ATP. This does not produce ATP. Okay, so wala siyang kinalaman kung well-fed ka ba or fasting ka ba. But you need to know this pathway. So this pathway is important for the following reasons. Number one, it is important because it produces NADPH. Now, NADPH is similar to NADH, but they're different. So, later we will show you para saan ba yung NADPH, nicotinamide adenine dinucleotide phosphate. Number 2, it produces ribose 5-phosphate, which is required for the synthesis of your nucleotides. So later, whenever you see NADPH or ribose 5-phosphate, and then I'll ask you, saan galing yan? What will you answer? Pentose phosphate pathway. Okay? So yun lang yung tatandaan nyo. Pag nakakita kayo ng NADPH, tsaka ribose phosphate, or even deoxyribose, you must remember nang galing yan sa pentose phosphate pathway. And then, this pathway is important for the metabolism of 5-carbon sugars. So napansin nyo kanina, no, puro 6-carbon sugars yung metabolize natin. Glucose, fructose, galactose. So here in the pentose phosphate pathway, we will metabolize 5-carbon sugars or pentose. It neither produces nor consumes ATP. So where does it occur? So the pentose phosphate pathway occurs in red blood cells and tissues that produce lipids. So for example, your liver, your adipose tissue, adrenal glands, thyroid, testes, and lactating mammary glands. So remember your adrenal glands, diba? Your adrenal glands, we produce there your steroids, no? So your mineralocorticoids and your glucocorticoids, no? Lipids yung mga yon. The liver and adipose tissue, they produce fatty acids. And your lactating mammary glands, they produce short and medium chain fatty acids for your breast milk. So the pentose phosphate pathway occurs in the cytosol. The substrate or the starting point, it is glucose 6-phosphate. Kaya nga ang alternative niya ay pentose phosphate pathway. So bakit siya tinawag na pentose phosphate pathway or hexose monophosphate siya? So remember, di ba, starting point natin glucose sa glycolysis. Yung glucose, kino-convert natin to glucose 6-phosphate. No. So glucose 6-phosphate. Okay? So instead na bumaba tayo to glycolysis, no? So instead na bumaba tayo sa glycolysis, anong gagawin natin? Nanakawin natin siya. Dadali natin siya sa hexose monophosphate shunt. Kaya siya tinawag na shunt. So you divert the glucose 6-phosphate, no, to the pentose phosphate pathway. So hexose monophosphate shunt. So kaya ang starting point niya glucose 6-phosphate. The important products, as I said earlier, will be NADPH and ribose 5-phosphate. So tatandaan na, NADPH, ribose phosphate, daling yan sa pentose phosphate pathway. The rate-limiting step is the conversion of glucose 6-phosphate to 6-phosphogluconate. The enzyme is glucose 6-phosphate dehydrogenase. So glucose 6-phosphate dehydrogenase, yung G6PD. Doon sa G6PD deficiency, yan yun. No? Yun yung G6PD sa G6PD deficiency. So here's what you need to remember. You must remember that the pentose phosphate pathway has 2 phases. A first phase and a second phase. And these phases have opposite characteristics. Yun lang yung kailangan nyo matandaan. The first phase, it's oxidative, irreversible. Baliktad naman yung second phase. Non-oxidative, reversible. Ulit, the first phase is oxidative, irreversible. The second phase is non-oxidative, reversible. Okay, so, paano yun? So, yung first phase, the important enzyme is your G6PD, glucose 6-phosphate dehydrogenase. So, first phase natin na po-produce yung NADPH. Sa second phase naman, the enzymes are collectively known as transketolases. You also have transaldolases there, no? But you have the transketolases, and then the important product here will be your ribose phosphate. So let's take a look. So in here, take note. So this is the first phase. In the first phase, yung glucose 6-phosphate natin, ino-oxidize. So using glucose 6-phosphate dehydrogenase, it is oxidized to form ribose 5-phosphate. So when we say oxidize, no? Tatanggalan natin, dadagdagan natin siya ng oxygen. Dadagdagan natin siya ng oxygen. And you will see it produces NADPH. Okay? it produces 2 molecules of NADPH. So first phase, oxidative. Irreversible. Kasi nga redox reaction siya, hindi siya reversible. No? Hindi siya reversible. Okay? The important product here, NADPH. So at the end of the first phase, you will see yung glucose 6-phosphate natin na 6 carbons, magiging ribulose 5-phosphate. 5 carbons na lang. No? Nag-release tayo ng carbon dioxide. No? Nag-release tayo ng carbon dioxide. We now go to the second phase. So itong second phase, exciting. No? So itong second phase, you will see this diagram. Look at this drawing. No? Pay attention to this drawing. So you will see here, no? In this drawing, may mga drawing ng blocks tsaka phosphate. So bawat isa dyan, ribulose phosphate. No? Ribulose 5-phosphate. So you see 5 squares kasi nga 5 carbons with the phosphate. No? So ito, yung second phase. Anong sabi natin? Non-oxidative, reversible. Ang analogy ko dito, para ka lang naglalaro ng Lego. No? Meron kang 5 blocks and 5 blocks. Yung 5 plus 5, anong pwede mong gawin? Pwede mo silang ipag-combine. Gagawin mo 7 plus 3. 7 plus 3. Yung 7 plus 3 na Lego, anong pwede mong gawin? I-jumble mo ulit siya. Gawin mo naman siyang 6 plus 4. So, para ka lang nagpapalit-palit ng Lego blocks, no? Sa second phase, no? So, 5 plus 5, gawin mong 7 plus 3, gawin mong 6 plus 4. Kaya you will see, no, maraming mga arrows. So, the enzymes that do this are collectively known as transketolases. And your transketolases require thiamine, which is vitamin B1, no? Vitamin B1. In the process, what happens, no? In the process, what happens? You generate carbohydrates with 3 carbons, 4 carbons, 5 carbons, 6 carbons, 7 carbons. So, jumble. Kaya, reversible, non-oxidative. Once again, what enzyme is important? Transketolases, which require thiamine. So that's why one way to diagnose thiamine deficiency is using your RBC transketolase assay. They look at whether your red blood cells have transketolase activity. Pag walang transketolase activity, ibig sabihin they're deficient in vitamin B1. So bakit nga ba ulit siya important? Once again, the pentose phosphate pathway is important for you to produce NADPH. So this is NADPH shown here on the left. What are the functions of NADPH? Number 1, reductive biosynthesis. So later, when we discuss fatty acid synthesis, cholesterol synthesis, you will need NADPH. No? Si NADPH na to manggagaling sa pentose phosphate pathway. Number 2, to reduce glutathione. Inside red blood cells, we need NADPH. No? To reduce glutathione, papakita natin ito later. Number 3, cytochrome P450. In the metabolism of drugs, so in pharmacology, you will discuss cytochrome P450. They will require NADPH. Bactericidal mechanism of white blood cells. I will show you this later. Your white blood cells, to be able to kill bacteria using oxygen-free radicals, you need NADPH oxidase. And then finally, to synthesize nitric oxide, which is the most potent endogenous vasodilator, you need NADPH. So here, this is glutathione. So look at glutathione. Glutathione consists of 3 amino acids, glycine, cysteine, and glutamate. No? Glycine, cysteine, and glutamate. So reduced glutathione, ito yon, GSH. Oxidized glutathione, ito yung magkakatabi, GSSG. No? So remember this, the 3 amino acids that make up glutathione. Tinatanong nila to. So you will see here, bakit important yung glutathione? So yung glutathione, hindi siya lang pampaputi. No? The more important function of glutathione is, it's an antioxidant. Ano ibig sabihin ng antioxidant? So you remember this, hydrogen peroxide. So hydrogen peroxide is a source of free radicals. Ayaw natin sa kanya. No? You don't want hydrogen peroxide. So to reduce hydrogen peroxide, to convert it back to water, no? So kailangan natin siyang, no, ma-reduce. So to reduce it to water, kailangan may ma-oxidize. So sino yung ma-oxidize? Si glutathione. So reduced glutathione, it is oxidized. At the same time, hydrogen peroxide is reduced. And the enzyme that does this is glutathione peroxidase. kung marami pang hydrogen peroxide, anong kailangan mangyari? Kailangan constant yung supply ng reduced glutathione, correct? So, paano natin ma-regenerate si reduced glutathione? So, the oxidized glutathione, it must be reduced using glutathione reductase. Okay? Pero kailangan kung ma-reduce siya, dapat may ma-oxidize. Sino naman yon? Si NADPH. Ibig sabihin, kailangan natin ng constant supply ng NADPH para palagi din tayong may reduced glutathione. Kung walang NADPH, what will happen? Kung walang NADPH, walang reduced glutathione. Kung walang reduced glutathione, you will not be able to reduce hydrogen peroxide. And therefore, you will have accumulation of free radicals. So once again, glutathione, you reduce glutathione is important to remove hydrogen peroxide. This is catalyzed by glutathione peroxidase, which requires, take note of this, selenium. So heavy metal selenium is needed to reduce hydrogen peroxide to water, convert reduce glutathione to oxidized glutathione. So saan ngayon manggagaling si NADPH? Saan ngayon manggagaling? So para meron tayong continuous supply ng reduced glutathione, saan manggagaling si NADPH? Sa pentose phosphate pathway. Okay? So sige nga, which phase? First phase or second phase? So NADPH is produced in the first phase, no? It's in the first phase. Anong characteristics ng first phase? Oxidative, irreversible. Oxidative, irreversible. What's the important enzyme? Glucose-6-phosphate dehydrogenase. So paano kung wala tayong glucose-6-phosphate dehydrogenase? Yun yung G6PD deficiency. G6PD deficiency is the most common disease-producing enzyme abnormality in humans. No? About 400 million affected individuals worldwide. So what will happen? If we do not have G6PD, hindi tayo makakaproduce ng NADPH. So sinong kawawa dito? Red blood cells. Why? Remember, your red blood cells, they carry oxygen, no? So sila yung prone to damage from free radicals of oxygen. So during oxidative stress, no? Meaning to say, during oxidative stress, like infection, no? Your red blood cells will not be able to defend themselves against oxidizing agents. So you see here, no? Normally, your pentose phosphate pathway will produce NADPH, and that gives you a constant supply of reduced glutathione. Kung walang NADPH, walang reduced glutathione. No? Kung walang reduced glutathione, what will happen? You see this, the free radicals. They will damage lipids, proteins, DNA, and pinakakawawa doon, red blood cells. So if you have oxidative stress, just as infection, or if you have drugs, no, may mga drugs that will trigger formation of free radicals, then you have accumulation of free radicals. No? You have accumulation of free radicals. That's because of G6PD deficiency. Kasi kung walang G6PD, walang NADPH, walang reduced glutathione, makakadamage ang tissue yung free radicals. So here, this is the manifestation. You will have hemolysis. No? So, kunyari, ang history nito, these patients will have hemolysis after oxidative stress. Most common infection. But then, other causes of hemolysis will be drugs, specifically your sulfonamides, cotrimoxazole, primaquine, chloramphenicol, and then fabamines. No? So you see this? These are what you call bite cells. No? And then these are Heinz bodies, which are hemoglobin precipitates. Okay? So always remember this, no? Heinz bodies and bite cells, no? Features of glucose-6-phosphate dehydrogenase deficiency. Saan may fava beans? Sa ding-dong. No? So your fava beans have ding-dong. So anong ginagawa sa mga patients with G6PD deficiency? You just advise them, no? So you must remember, you must give them advice na, ito yung mga triggers natin for hemolysis. Because you don't want them hemolyzing every so often. Kasi ayaw natin yun, no? They will become anemic, they will have jaundice, they can have end organ damage from iron accumulation, no? Ayaw din natin sila laging tinatransfuse. Okay? So, you advise the parents. So, kunyari, ikaw, alam mo may G6PD deficiency ka, huwag mo siya resetahan ng cotrimoxazole, no? If your patient has G6PD deficiency, hindi siya pwede mag-cotrite. Then finally, chronic granulomatous disease. Sabi natin kanina, no? Your free radicals, they're not always bad. So, in here, yung neutrophil. Kunyari, neutrophil, kumain siya ng bacteria. No? Kinain niya yung bacterium. Yung neutrophil, yung oxygen, i-convert na to superoxide. Okay? So, superoxide, and then hydrogen peroxide, you will see, hey, that is used to kill the bacterium. But to be able to do that, no, to convert oxygen to superoxide, it needs the enzyme NADPH oxidase. No? NADPH oxidase. So, paano ngayon kung walang NADPH oxidase? If you don't have NADPH oxidase, you will not be able to create this, no, the superoxide. So therefore, yung neutrophils, kain sila ng kain ng bacteria pero hindi nila napapatay. They will just wall off the infection using with granuloma. So nawo-wall off lang infection. No? So they will present with severe, persistent, chronic pyogenic infection. So paulit-ulit na abscess. Okay? Paulit-ulit na abscess. That's chronic granulomatous disease. NADPH oxidase deficiency. So in summary, no? So let's summarize. Glycogenesis is the synthesis of glycogen, which is the storage form of glucose, whereas glycolysis is the retrieval form of glucose from glycogen. So remember, glycogenesis, glycogenolysis, both of them occur in the liver and muscle kasi walang ni. So what is activated when you're well-fed? Glycogenesis. You want to store glucose as glycogen for future use. What is activated when you're fasting? Glycogenolysis. Because you're fasting, you want to synthesize glucose that can be used by tissues. What is the end product of glycogenolysis in the liver? Free glucose, because you want to distribute glucose to the tissues. What is the end product of glycogenolysis in the muscle? It's glucose 6-phosphate, because muscle stores glycogen for its own use. So enzyme deficiencies in glycogenolysis result in glycogen storage diseases. So you must understand, no? Bakit ganun yung manifestation ng von Gerk disease? Bakit ganun yung manifestation ng Pompe disease? Bakit kapag McArdle syndrome walang hypoglycemia? So please review this. So in the exam, you will usually be given a case, and then sometimes they just ask you the diagnosis. But most important, you need to understand why. Enzyme deficiencies in the metabolism of galactose and fructose may cause benign or severe symptoms. So what will cause benign symptoms? If the deficiency is enzyme number 1, the first enzyme. So that's either galactokinase or fructokinase. What will cause severe symptoms? If the enzyme deficiency is step number 2. So that's aldolase B, resulting to fructose intolerance, or galactose 1-phosphate uridyl transferase deficiency, resulting in classic galactosemia. So, how do we treat these conditions? Avoid fructose or galactose. Avoid sucrose or lactose. Complications of diabetes arise from the accumulation of sorbitol in the tissues. So remember, glucose, when you reduce it, it becomes sorbitol. No? Sorbitol is a product of reduction of glucose. In diabetes mellitus, excess sorbitol attracts water, causing end organ damage in DM. Although it does not produce ATP, the pentose phosphate pathway is important for the synthesis of NADPH and ribose 5-phosphate. So remember, anong difference ng phase 1 or first phase? Anong importance ng phase 2? The first phase is oxidative, irreversible. The second phase is non-oxidative, reversible. Okay. G6PD deficiency leads to a decrease in NADPH, no? Presenting with hemolytic anemia after oxidative stress. So remember, anong role ni reduced glutathione, no? So you must understand how it helps prevent the accumulation of oxygen-free radicals. Thank you for listening to this lecture.